Hi everyone, I am Dr. Savetha. I am working as assistant professor in Tagore Medical College, Chennai. Before I am going to section, I really want to thank Dr. Raghunandan, who is the founder of this Medusane, and he is doing a wonderful job and he worked hard for this. I am really proud of him, and I want to congratulate him for a 6.3K subscriber. All the best, Rahul. Okay. Today, I'm going to discuss about the mycology part. Initially, I have I have divided into part 1 and part 2. So in part 1, I'm going to discuss about the introduction, superficial and the subcutaneous mycosis. Okay. Today we go into the part 1 section. So we go into the section now. Okay. So what is the medical mycology first? What is medical mycology? It is nothing but dealing with the medically important fungi. So, I'm going to cover like the classification of the fungus, laboratory diagnosis of the fungal infection, the treatment of the fungal infection. So, this main fungus actually is derived from the Greek word called myx, meaning mushroom. Okay. So, coming to the classification, actually, the classification of the fungi is broadly classified into three types based upon the morphology, based upon the taxonomy, and based upon the fungal disease is produced. So, first we'll see about the morphology of the morphological classification. First one is yeast. What is yeast? Yeast is nothing but the round to oval cells that reproduce by budding. Actually, after this reproducing, it is separated from the parent cells. Okay, that is the yeast. For example, for the yeast is a Cryptococcus neoformans and Saccharomyces cerevisiae. So, in Cryptococcus neoformans are pathogenic and the other one is a non-pathogenic. Coming to the second one is a yeast-like. Is yeast-like is a yeast which is forming along with the pseudo-hyphae. The example is a Candida. Here, after the forming of the yeast cells, which is not separated from the parent cell, which is attached to the parent cell. Okay, that's why it's called as a yeast-like. Okay, and it have how to differentiate from the true hypha means it has a constitution of the septa. So this is the constitutions of the septa. Okay. Coming to the third one is the mold. What is a mold? Mold is also called as a hyphae. So in the top picture, so this in this picture you can able to see this is a septate hyphae and this is the non-septate hyphae. In the septate hyphae there is a transverse line and the non-septate hyphae there is no any transverse line. Okay, then coming to the based upon the growth pattern in the culture medium, it is broadly classified into aerial mycelium and the vegetative mycelium. So aerial mycelium is nothing but a part of the mycelium which project above the surface of the culture medium. Vegetative mycelium which is a that grows on the surface of the culture medium. Okay. Then fourth one is very important. It is a dimorphic fungi. Dimorphic fungi is nothing but the fungi which is grow in the two ambient temperature. One is 25 degrees Celsius and another one is 37 degrees Celsius. In 27 degrees Celsius, the fungus exists in the mold form. Then at the 37 degrees Celsius, it exists in the yeast form. Okay, so dimorphic fungi is a very important short note in the PG as well as in the UG students. What is the fungi which come under dimorphic fungi is all the systemic fungus, that is a Cystoplasma capsulatum, Blastomyces dermatitis, Coccidioides fumitoris, Paracocidus brasilanus, Penicillium aeruginosa, and Sporosis shantai. I tried to explain about this dimorphic fungi in part 2 in detail. Coming to the taxonomical classification, so in the taxonomical classification based upon the sexual spores, okay, the first one is a Zygomycota, which is a sexual spores form which is called as a zygospores, and it possesses aseptate hyphae. What is example for the Zygomycota is Rhizopus and Mucor. Coming to the phylum Ascomycota, and the sexual form spores form is called Ascospores, and it possesses the septate hyphae. Example for this is Aspergillus. Coming to the phylum Basidomycota, and this forms a sexual spores called Basidospores. Example is Cryptococcus. Fourth one is a Deuteromycota. It is also called as a fungi imperfecti. Its sexual state is either absent or unidentified. Then third one actually it is based upon the fungal disease. The fungal disease actually I want to explain in detail. That's why I didn't display this slide, but I will explain what is that. So based upon the fungal disease, we classifying it as a systemic disease and superficial, systemic, subcutaneous, and opportunistic. Okay, so under the under the superficial, mainly the fungus is infecting the skin, hair, and the nail, and subcutaneous it infects the subcutaneous tissues and the bone. Systemic, it is mainly formed by the inhalation of the spores is while entering to the lung. From the lung, it is disseminated into various parts of the organs. It causes a systemic disseminated systemic infection. Okay. Then coming to the opportunity fungal infection is mainly occurred in the patients who have a lower immunity. So while I am explaining about each one in a separate, I will explain what is the classification and what are the agent comes in that diseases. Okay. Then coming to the type of this fungal spores, there is a sexual spores and asexual spores. Under the sexual spores, there is a zygospores which is observed in the Mycomycetes, ascospores which is observed in the Aspergillus, basidospores which occur in the Cryptococcus. Then coming to the asexual spores, there is a vegetative asexual spores and the aerial asexual spores. In vegetative asexual spores, there is arthrospore, blastospore, and femtospore. And aerial asexual spores are conidiospore, sporangiospore, and microconidia and macroconidia, which is observed in the dermatophytes. Next is this is a they may ask us a separate question like write about the laboratory diagnosis of the fungus disease. So if they ask in a 10 marks means like this only you have to cover. We'll see how to cover the laboratory diagnosis of the fungal disease. So before what is a lab diagnosis? We're going to identify the fungal diseases. Okay, so we want to start from the specimen collection. Sorry. Excuse me. Okay. So specimen collection, what are the types of the specimen you get for the fungal infection? Means you get the skin, hair, nail, sputum, pus, grains. Okay. And for systemic mycosis, if you're suspecting this, we want to take a blood sample of the CSF, etc. Okay. After collecting the specimen, you have to transport it into to the laboratory immediately as soon as possible, and they want to process into the primary media immediately. If there is any delay in sending the sample to the laboratory means how you want to transport it. Okay, you want to store the specimens usually at the temperature of the 4 degrees Celsius. Usually all the fungals will be maintained at the lower temperature. Okay, except the blood and the CSF should be stored at the 30 to 37 degrees Celsius, and all your dermatological specimens. What is called as a dermatological specimen means this fungal is called a dermatophytes. It's called as a dermatological specimens which is stored in the 15 to 30 degrees Celsius. Okay, then we have stored we have processed the specimen. Then we want to go into process the specimen. If after receiving the specimen, some specimen we can able to directly inoculate it into the media. For example, abscess aspirate, bone marrow aspirate, CSF, swabs, body fluid, hair, skin scraping, nail, bronchial washing, brushings, etc. can be directly inoculated. Some require a special concentration before inoculating into the media. For example, if you receive a sputum sample, means you want to treat it with the N-acetyl L-cysteine. For blood sample, you want to collect in the biphasic medium in the two bottles, and it's maintained at the two different temperature because the blood sample usually caused by the systemic mycosis, right? So in the systemic mycosis, I have already told you that mostly causes the dimorphic fungi. So it may grow at the 2 ambient temperature. So we want to inoculate that. We want to maintain the blood culture bottle in the 2 ambient temperature. Okay. Then coming to the body fluid and the urine, before inoculating in the specimen, you want to filtrate and centrifuge the sample. As well for that, like that only for the exudate pus and the grains. If you are receiving a grain sample from the mycetoma, means you want to crush the grains and then only you want to inoculate in the media. Then CSF usually you want to collect the 10 to 30 ml. CSF sample also before inoculating into the media, you want to centrifuge the sample. In the centrifuge, during the centrifuge, we will have a sediment. We will get the sediment. The sediment part is used for the culture and the microscopic identification, and the supernatant part is used for the antigen detection. Then coming to this, after we got this specimen, we have transported and we have processed. Okay, now what we want to do means we want to do the microscopic examination. First, we want to, after receiving, like in bacteriology, receiving sample, we immediately do the gram stain and see, right? Like that, we want to immediately do the, for fungus, we want to do the KOH mount. What is the call as a KOH mount? Is nothing but the potassium hydroxide. Okay, so usually the concentration we used here is a 10% for the all the sample, except the sample like nail and the biopsy is required required a 20 to 40% because it have a thick in consistency. We want to dissolve the KOH should be dissolve the keratinized tissue, so that we have to increase the concentration of the KOH. Take a test tube and pour the 20 to 40% of the KOH and immerse the specimen and keep it at overnight incubation, and next day only you have to look for the KOH mount. Okay. So this is the picture. Here we can able to see the hyphae and the yeast cells. Okay. Then coming to the other microscopic findings with the gram stain. I think you are most familiar with this gram stain procedure and the all steps and what are the stains used in gram stain. Which is mainly gram stain is used to identify the Cryptococcus and the Candida. So in this picture we can able to see the yeast cells and the hyphae. This is a pseudo hyphae which all take up the gram positive. Okay, and another stain, important stain is the India ink and the Nigrosin stain. And this is also used to demonstrate the Cryptococcus, the capsule of the Cryptococcus. For this, actually, we want to take the specimen like a CSF, body fluid, urine, whatever in a direct slide, and to put a drop of the India ink and put a cover slip. We want to view under the microscope, and the that stain should not be in black color. It should be normal brownish in color. If suppose it is too dark, the stain is very too dark, we have to add 2 to 3 drop of the distilled water to it and then examine. Okay. Then coming to the histopathological stain is mainly used for the biopsy tissues. So what are the histopathological stains used to identify the fungus? It means the periodic acid shift stain and the gomeramine methylamine sylvane is most commonly used. So periodic acid shift stain is usually stains the fungus into magenta or deep pink in color, and is mainly stains the live fungi. So this gomeramine methylamine sylvane is an alternate for the PES. Why? Because it has able to stain even the dead fungi also. Okay, that is the main difference between both this stain. And in the gomeramine methylamine sylvane, the background will be in green color, only the fungus will take up the black color. And there is other stains like Myosine phantomone stain and Hematoxylin eosine stain and the Myositis stain also. So these are the features for this histopathological stain. This is the periodic acid shift stain and the Myosine phantomone stain and the Hematoxylin eosine stain, Myositis stain, and the gomeramine methylamine sylvane stain. Next important microscopic identification with the stain called the lactophenone cotton blue, also called as the LTCB. Okay, so each one is a stand for have a specific function. So L is a stand for lactic acid and does have a function to preserve the morphology of the fungi, and B stands for the phenol and which is disinfect, act as a disinfectant, and the CB is a cotton blue, is nothing but to stain the fungus. Okay, and along with that we have added the glycerol also to prevent the draining. So this is the exact picture of the LCBCB mount. Okay. Then after specimen process, transport and direct microscopic examination, we want to culture the specimen, right? So what are the basal media we use for the fungus means Sobrus disturbs agar. That is a basal media we use for the fungal cultivation. If suppose during the fungal cultivation there will be an interference of the other bacterial growth also. So how will you inhibit that? While adding the antibodies to the SDA, it will become a special media so that we can inhibit the other bacterial growth. Okay, other than the SDA, there is a convened agar also which is used to cultivate the chlamydia spore production. Okay. So, other than this convened agar and SDA, there is also the brain heart infusion agar and the Niger seed agar, which is used both are used for the Cryptococcus. And there is like in the bacteriology we use the McConkey agar, right? Like that, here we use the chrome agar for differentiating the medium, act as a differential medium. Is mainly used for the speciation of the Candida. Can you able to see the different colors here? So this is the different species of the Candida. Okay. So, actually, the fungus should be maintained at an exact temperature. It's not like a bacteriology. We want to maintain at a lower temperature. What is the temperature? 25 to 38 degrees Celsius. And also, for the in case of the suspecting the dimorphic fungus, you want to maintain at the 2 ambient temperature, right? Okay. Then there is a special incubator called the BOD incubator, which is also called as a biological oxygen demand incubator, which maintains the fungus at a lower temperature. So, and also then all the fungus will take around 2 to 3 weeks to grow. Okay, only the saprophytic yeast and the other opportunity fungus will grow within 5 days, but all the rest of the fungus will take around 2 to 3 weeks. Okay. So after you're culturing, after 2 to 3 weeks, you're taking the test tubes and you want to look for what are all points you look for the macroscopic identification. First one, you want to see the rate of the growth. If you have taken the test tube within 5 days, if you are suspecting the saprophytic or aprocytic or any yeast you have inoculated, means it should be grown within 5 days. So rate of the growth. If suppose you are suspecting the dermatophytes, means it should grow, and the growth will start after the 1 week only. So after 2 weeks, you want to look for the pigmentation or the texture, whatever it is. So first one thing you want to note is the rate of the growth, and second one is a pigmentation. So after taking the test tube, you want to turn the back side of the test tube, reverse. Okay, reverse side of the test tube. Reverse it means there will be a pigmentation, like this color changes. Okay, so you can able to see here. This is a mild orange in color. It's a dark red in color, and as per the Niger will form as a black in color. So this pigmentation will give idea about the what fungal species has grown. Okay. Then coming to the third one is the texture. So why the macroscopic appearance? We can able to see whether the colonies is a smoothy, waxy, powdery, cottony. So we want to explain about the texture of the colonies also. Okay. Coming to the next is microscopic appearance of the fungi. So how we are going to find out the microscopic appearance of the fungi? In bacteriology, what we will do, from the culture plate, you will take a loop of the colony and look for the and staying with the gram stone and look for the microscopic fungi. Here also what we are doing means we want to take the colony from the culture. So how we take the fungal material from the culture media means we can't able to touch with the loop of the straight wire. We need two needles, that is called as a teasing needles. We call it as a teasing needles. We want to hold it in both the hands, and like we would with the help of the needle, we want to take this part of the fungal elements, I mean fungal specimens, and put it into the slide and put a drop of the LCB mount and put a cover slip and view under a microscope. So this is called as a tease mount identification method. And there is a slide culture method. In slide culture method, what we want to do means we want to take a that is agar block of SBA agar block around 1 centimeter. 2 blocks we want to take. Okay, and place at the corner of the slide, then and place it over the blended glass rod. Okay. In the above the agar blocks, we want to streak this fungal specimen from the stalk culture and the other at the edges of the agar block, and put a cover slip, and this region you have to dip cotton in the water and place it here and cover the petri dish and incubate it overnight. Okay, the next day or leave it for next day or 2 days, 1 day after. 2 days after, you take this cover slip. You want to take the cover slip from this agar block. Over the cover slip, put a drop of the LCB mount and look for the microscopic fungi. Okay, like this only you want to see the microscopic appearance of the fungi. Okay, and there is a cellophane tape mount also. That is, after taking the cover slip, some of the elements will be present in the agar block. Okay, so with the help of the cellophane tape, slowly touch the fungal element and reverse the cellophane tape and put a drop of the LCB mount over it and put a cover slip and view under the microscope. Okay, that is called a cellophane tape mount. So what are the other method other than this means is for Candida we have a germ tube test and dermal plate culture, and for Candida we have do a sugar assimilation test. Okay, for dermatophytes there is a hair perforation test, and urease test usually we do for the cryptococcus. These are the other method of identification of the fungus. Then coming to the immunological method, what are the immunological method we do is antibody detection by using ELISA and immunodeficiency test and the complement fixation test, antigen detection by the lattice agglutination test for cryptococcal antigen, immunochemistry also for detecting the antigens. Then there is automated culture also we can do, automated method also, diagnostic method also we use by using the MALDI-TOF and the Vitek. It is now recent advance in the microbiology. Coming to the molecular method, we do the real-time RT-PCR and the DNA sequencing method. What is the treatment of the fungal infection? The treatment of the fungal infection is like we use in the amphotericin B is mainly used for systemic mycosis, which comes under pollens. Then echinocandins you use for the systemic mycosis, and the benzoflurans is used for the dermatophytes, that is like rhizofulvin. Okay. Next is azoles. Like azoles, like chlorotrimazoles, ketoconazole is a topical use, and fluconazole is for cryptococcus. Itraconazole is also used for the cryptococcus sporotrichia and all. Then antimetabolite flucytosins is used for the systemic mycosis. Allylamine is used for the terbinafine topical use. So here I am come with the end of the section of introduction part. Okay, now we will see about the superficial and the subcutaneous infections. Okay, coming to the superficial and the subcutaneous mycosis, first we see what are the superficial disease caused by the fungus and what are the subcutaneous diseases caused by the fungus. First, we see the superficial diseases like fungal disease called as a Tinea versicolor caused by the Mycology furfur, Tinea nigra caused by the Hortia wernicki, Pedra is caused by Trichosporidium bigli and Pedra horte, and dermatophytosis caused by Trichophyton, Microsporidium and Epidermophyton. These are the superficial mycosis. Okay, coming to the subcutaneous mycosis, what are the disease names? Mycetoma is caused by Medulla mycetomatosis, Pseudorachida boydi, and Sporotrichosis caused by Sporotrichia shakeni, and Chromoblastomycosis caused by the Phyllophora vercosa, and Rhinosporidosis is caused by the Rhinosporidium sibiri. So now I am going to explain only the important fungus which is come for the exam point of view. I am not talking about the unimportant fungus one which is non-pathogenic also. Okay, so only I am going to concentrate on pathogenic and important question for the examination. The first is superficial mycosis. What they may ask is the important shortener. What is the Tinea versicolor? It is also, they may also ask as a Pityriasis versicolor. What is mainly caused by the agent called Mycology furfur. Here what is happening is there will be a flat, round, that is hypo to hyperpigmentation on the skin. Usually is seen in the areas rich in the sebaceous gland like neck region, chest and the upper arms. Okay, it is usually a non-inflammatory and a non-puritic. Okay, there will be no itching in this lesion. So other than this, they will have, patient will have also have a seborrheic dermatitis. What is a seborrheic dermatitis? Is nothing but a dandruff. Okay, for the adult, and for the children it is called as a cradle cap. It is mainly, it is very severe in the AIDS patient. Then after that seborrheic dermatitis, they will also have a atopic dermatitis, folliculitis and disseminate also, but it's very rare condition. So how are you going to diagnose this? Samples, like we get a skin samples. So we want to first do the KOH mounting, and in the KOH mounting we have like this is the exact picture of this. Like it is a hyphae and this is the yeast cells that is called as a this appear as a spaghetti and meatball appearance. So while while you are attending this question, if you are writing about the laboratory diagnosis, means you want to be more specific in this point. You have to write this point so that only you get the full mark. Okay. Then coming to the lab culture, so we want to culture with SD with olive oil overlay it. The appearance of the colony is like there is a fried egg colony appearance after incubating for 5 to 7 days at 32 to 35 degrees Celsius. And urease test also we can able to do, which is a positive. And a wooden lamp, this is called as a wooden lamp. If you viewed skin lesion through the wooden lamp, it's fluorescent to the golden yellow fluorescence. Okay. So what is the treatment given for this Tinea versicolor is we give a topical lotion like selenium sulfate and ketoconazole cream also we can use. If suppose you have a dandruff, they have a dandruff means we can give a ketoconazole shampoo also and selenium sulfate shampoo also. Okay, then there is a terbinafine cream. All should be prescribed for the 2 weeks. Okay, for the effective treatment. Then what is a Tinea nigra? Tinea nigra also is a non-scaly patches present or mainly occur in the palm and the sole. Is most common seen in a females and caused with the Hortia wernicki. Okay. Coming to the Pedra. Pedra is a very important short note. If they are asking the short notes, you want to write both white Pedra and the black Pedra. Okay, what is a white Pedra means? In the hair shaft, it forms white color nodules which is less firmly attached, and it's mainly caused by the species called Trichosporidium bigli, and also the urease is positive for this organism. If you culture this organism, means it produces a white color colonies, and in a KOH mount we can see the hyaline septate hyphae with the Arthrospores is the rectangular arthrospores. This is the exact picture of the pedra, white pedra. Okay. Then coming to the black pedra. Black pedra is nothing but the black nodules which is firmly attached. That is less firmly attached. Here it is firmly attached. Okay, and there's a caused by the agent called Pedra hortae, and it's a pyoid fungus. What is a pyoid fungus? It's nothing but the pigment producing fungi. Okay. So in colonies like in it contains reddish forms a reddish brown colonies, and it in KOH mount it contains forms a dark brown thick septate hyphae seen with the ascus. This is ascus containing the ascospores. Next important one is the dermatophyte. Under the superficial mycosis, they may ask a dermatophyte as an essay. Okay, so dermatophyte is nothing but it's also called as a tinea, and also a ringworm. It is also called as a ringworm. Okay, it's the most common superficial mycosis which affect the skin, hair, and the nail. Okay, so there is dermatophyte has a three species: trichophyton species, mycosporum species, and epidermophyton species. In trichophyton species, all three is affected: skin, hair, and nail. And mycosporum species infect only the skin and hair, and epidermophyton species only is infect the skin and the nail. Okay, based upon dermatophytes is based upon classified based upon the habitat. So first, this is an anthrophilic. What is anthrophilic? It's nothing but it's infected the human. Okay, so what are the species infecting means in trichophyton is rubrum, antagrophytes, Cholini, transdens, and violation. Mycosporum, ordini, and epidermophyton, foloscum. Okay, zoophilic is nothing but the which is infection in the animals. It is mainly in the trichosporum species like Equinum vercosum and Mycosporum canis and equine. And geophilic, which is found in the environment, so Trichophyton ajoli, Microsporum egyptium, Epidermophyton stockdale. So how the lesion of the dermatophytes will look like? So here you can see the center clearing with outer is raised edge. So it looks like a ring. Okay, so that's why it's called as a ringworm infection. Okay, so called as a ringworm. So in nail is usually infect the superficial part and then its lateral side, then finally it's spread throughout the nail. And hair first essentially infect the hair shaft and it become into the alopecia. So clinical type of the dermatophyte, we call it as a tinea capitis. Tinea capitis has the following types: carrion, flavus, ectothrax, and endothrix. Carrion is nothing but the body lesion caused by vercosum, T vercosum, and flavus is a crust-like which is caused by the tree Cholini, and ectothrax is caused by the is an orthospore formation occur on the surface of the hair shaft, caused by the M. adoni and canis and T. antagrophytes. Endothrix orthospore formation occurs within the hair, completely filling the hair shaft, caused by the T. transference and the violation. Then tinea corporis, which is infect the non-hairy skin of the body, trunk, and the limbs. Tinea pedis with athletic fruit, okay, so between the web space it causes. Tinea curis is nothing but the groin area, and tinea barbae is infect the beard and mustache, and facie, which is occur in the face. And tinea imbricata, the concentric lesion of the skin. Okay, tinea concentrica. Angoium is caused by the nail, and manum is caused by the palmar aspects of the hands. So these are the ringworm infection. We can see the first picture is a tinea capitis, and this is the second one facies, third one pedis, and fourth one is the corporis. Laboratory diagnosis of this dermatophytosis. First, we do the Woodlam's examination, which is a positive for Microsporum species and a Trichophyton cholini. And this fluorescence is mainly due to the pigment produced in the cell wall. So, what are the specimen we get for the dermatophytosis means we get a skin, hair pluck. We should not break or we should not cut the hair. Okay, we want to pluck the hair, and nail clipping also used for the dermatophytic identification. While you put the KOH mode, we can see the septate hyaline hyphae, and this is a picture of the ectothrix, and this is the endothrix of the hair shaft. Okay. Then you are going to culture it in the SDA containing the cyclohexavan is nothing but antibiotic. Okay, and also we can cultivate it in the potato dextrose. So what are the macroscopic appearance? We want to look for the rate of the growth, texture, and the pigmentation. So these are the polymorphology of the dermatophytes. This is the first, which is a trichophyton, a microsporum, which is a red in color, and epidermophyton, which is from the picture itself we can able to see that it is in a cotton woolly appearance. Like that, we have to tell about the texture. Okay. Coming to the microscopic appearance, we will put the LPCBTs mount. We can able to see the hyphae and the spores. So spores we see in dermatophytes, we call it as a conidia. So there will be a macroconidia and the microconidia, and there is a special hyphae. I will explain the picture. So this is the macroconidia of the trichophyton species, which is a pencil shaped, and this is the macroconidia of the microsporum species, which is in a spindle in shape, and this is the macroconidia of the epidermophyton, which is in a club shape, and this is a microconidia of the trichophyton species, and some microsporum species also will have the microconidia, but it is rare, and in epidermophyton, the microconidia itself has. Okay, so this is the macroconidia and this is a microconidia. Okay, and this is the hyphae. This is spiral hyphae, and this is a racket, and this is a pavichander. So after LPCBT's mount, you can able to see here. No, there is a macroconidia and as well as a microconidia. In Microsporum candis, there is only a macroconidia, which is a spindle in shape, and epidermophyton also there is only a macroconidia, which is in a club shape. So I will explain the same thing only here I have displayed. So what are the other method of the diagnosis? Means there is a hair perforation test. After plucking the hair, put it in the petri dish, add some water, add some yeast extract, and close the petri dish and incubate at a lower temperature. The next day, we can able to see the wedges appearance in the hair shape, like wedge shape. Okay, so this is one of the tests, and we'll do the urine test, which is positive for the trichophyte and mentogrophytes, and dermatophyte and molecular methods like PCR and skin test also we can do with, because it can able to detect the trichophytin, which is hypersensitivity to the trichophytin. Okay. Treatment what we are going to give is terbinafine or itraconazole. Depending upon the site they infect, we can give it for skin lesion we want to give for 1 to 2 weeks. For hair we want to give for 6 weeks. For nail we want to give for the 3 months. Okay. Alternatively, you can also give the oral isoflavin and the ketoconazole. For topical lotion we can give the Whitefield ointment. Next, we entering into the subcutaneous mycosis. The subcutaneous mycosis is mainly which is occur in this type of the fungus which is inhabited in the soil. So that's why the subcutaneous how it enter into the body means through the thorn tick or any splint injury, it will enter into the human body. Okay, and this produce a granulomatous lesion. So under the subcutaneous mycosis, the important one is a myosarcoma. What is myosarcoma? Is a very, very important essay. It causes chronic, slowly progressive granulomatous infection in the skin and the subcutaneous tissue, and it forms a triad of the infection. What does that mean? It forms a swelling, and the patient will have a discharge sinus. In discharge sinus, there will be presence of the granules. This myosarcoma is also called as a Madura mycosis or Madura foot. Why? Because means which is described in Madurai. First, they described in Madurai, South India by the John Gill in 1842. Okay. So, type of the mycetoma and the causative agent is first mycetoma is caused by the fungi, and actinomycetoma is caused by the bacteria, and there is a vitreomycosis which is caused by the Staphylococcus aureus. So, what is the main difference between eumycetoma and actinomycetoma? Is eumycetoma which causes black granules and white granules, and actinomycetoma which causes white to yellow granules and pink to red granules. Okay. Coming to the pathogenesis, I have already told this mainly occurred by the thorn tick. Okay, and is enter into the skin and causes a subcutaneous infection and causes granulomatous disease. So this is what I have told as a triad. So that we can able to see here it's a swelling, register, sinuses, and granules. I can't able to appreciate here, but there will be a discharge of the granules also. Okay, and sometimes it may enter into the bony lesion and causes the osteolytic and the osteoclerotic bony lesions also. Usually it is a painless. Okay. So in eumycetoma and versus actinomycetoma, the tumor caused by eumycetoma is a single well-defined margin, and actinomycetoma is a multiple and ill-defined margin. Sinuses appears late here, it appears early here. Discharge is serous, and here it is a purulent. Grains is a black to white in eumycetoma, here white to red. Bones involvement in osteoclerotic lesion, here is osteolytic lesion. Grains contains a fungal hyphae, here it is a filamentous bacteria. Laboratory diagnosis of the this mycetoma is mainly we collect these grains. We want to trace the grains between the slice and view under the microscope. Okay, macroscopic appearance, we want to look for the color, size, shape, and structure of the colonies. In microscopic, if you are suspecting the mycetomas, we will put the KOH mount. On the KOH mount, we can see the hyphae with the like there is some like some yeast depots. And if you are suspecting the actinomycetoma, you have to perform the gram stain, which is here we can able to see the gram positive hyphae here. And also we want to do the modified acid fast staining because the Nocardia is a mild weakly acid fast. Okay. Then histopathological stain we use for mycetoma, which is a granulomatous situation with a palisade arrangement of hyphae in a cement substance. So here we can able to see the edges which is very clear here. Here this is the granulomatous arrangement of filamentous bacteria at the margin. We can able to see the bacteria at the core which is radiating. Okay. Culture, if you are suspecting a mycetoma, if you want to inoculate an SDA, if you are suspecting an actinomycetoma, you want to inoculate it into the bacteriological media. So you want to culture both in SDA as well as a bacteriological media, like Elgin medium and the blood agar. Okay. Treatment, what a treatment you're going to do, this was surgical treatment is only treatment, and if you're suspecting mycetoma, you want to give the antifungal like itraconazole or amphotericin B for 8 to 24 months, and for actinomycetoma, you want to give a Welch regimen. What is that? Nothing but amikacin plus chlorpromazine. Next important subcutaneous microsis is sporotrichosis. Is also known as a rose garner's disease, and is mainly caused by the Sporoscia shekhentai, and is mainly spread via the lymphatics. Okay, so what other type like they causes osteoarticular type seen in among the alcoholic, and pulmonary type in the COPD, and disseminated sporotrichosis in the immunocompromised patient. Okay, fixed cutaneous type means a single nodule which is found without the lymphatic spread. Laboratory diagnosis, Sporoscia shekhentai actually it is a dimorphic fungi, right? Okay, you want to note down that point. In specimen we can get like first aspirate form nodules, curettage, and swabbing from the others. KOH mount you want to do. In the KOH mount we can able to see the hyphae and the yeast cells. Okay, it forms a flower pattern. And the histopathological stain if you do means they will form the astroid body. What is the astroid body means? This is a yeast cell surrounded by the eosinophilic mass and antigen antibody complex. Okay, it's also called as a Flendel-Hofli phenomena. So if you are writing about this sporotric, they are asking a question about sporotric means if you are writing laboratory diagnosis. In this laboratory diagnosis, histopathological stain, you want to mention about this asteroid body and the Flendel-Hofli phenomena and draw the diagram. So like this only you have to able to score the mark. Okay. Then your culture it is most definitely true for a diagnosis, inoculate an SDA and as well as a blood agar. So because it is a dimorphic fungi, we want to maintain at a true temperature 25 degrees Celsius and 35 degrees Celsius. So this is in 25 degrees Celsius forms a mycelial form, and then 37 degrees forms a yeast form. Okay, so this mycelial form, this is a flower pattern. It forms a flower pattern, flower-like pattern. Okay. So what are the other identification by the serological test? Skin test, because it is sensitive for the sporotrichine antigen. Okay, and what is the treatment we can give? Itraconazole drug of the choice, and duration for the 2 to 4 weeks. Okay. Then coming to the last one is a rhinosporidiosis. Rhinosporidiosis actually it is forms a large febrile polyp on the nose, and also it forms in the conjunctiva, and also we can see in ears, larynx, bronchus, and the genitalia also. What is the agent caused with the rhinosporidiosis? Rhinosporidium febrae, which is actually present in the aquatic area. So that's why who swims in the contaminated ponds, they will get this rhinosporidiosis infection. Okay. It is mainly actually the source is found in the Tamil Nadu, Kerala, Odisha, and Andhra Pradesh in India. So this is a histopathological polyp. This is a spirals. We can able to see the spirals, right? Okay, stain better with the muscaramin also, and the Rhinosporidium separate we cannot able to cultivate. So what treatment we can give is a radical surgery with the cartilage also mainstay of the treatment. Dapsone also we can give. But only thing is the recurrence is very common in the rhinosporidiosis. I think I'm clear with the introduction and the superficial and subcutaneous mycology part. I will see you soon with the part 2 mycology. Thank you and all the best.